Hello, welcome to World of Microbes. Today, we delve into the blueprint of life itself, genetics. This foundational science not only explains why we resemble our parents, but also drives modern medicine, agriculture, and our understanding of evolution. Genetics is the scientific study of heredity, genes, and genetic variation in living organisms. It explains how traits, such as eye color, height, and susceptibility to diseases, are passed down from parents to their offspring. It examines how biological information is stored in DNA and influences everything from physical traits to disease risks. Genetics forms one of the central pillars of biology and overlaps with many other areas, such as agriculture, medicine, and biotechnology. Genetics is the biological study of heredity and variation. It revolves around DNA, deoxyribonucleic acid, which is the molecule containing the instructions an organism needs to develop, live, and reproduce. Mastering a few key concepts provides a strong foundation for understanding genetic science. Fundamental building blocks. DNA, the double helix molecule that holds your biological code. Genes, specific segments of DNA that serve as the basic unit of heredity. They provide the instructions for making proteins, which perform essential functions in the body. Chromosomes, thread-like, coiled structures inside the cell nucleus made of tightly packed DNA and proteins. Humans typically have 23 pairs, 46 total, of chromosomes. Genome, the complete set of genetic instructions, or DNA within an organism's cell. Inheritance and variations. Alleles, different, alternative forms or versions of a specific gene. You inherit 2 alleles for every gene, one from each biological parent. Genotype, the exact genetic makeup or specific combination of alleles an individual carries. Phenotype, the observable, physical traits of an organism, e.g., eye color, height, or blood type, resulting from the interaction between their genotype and environment. Dominant versus recessive. A dominant allele requires only one copy to be visibly expressed in the phenotype. A recessive allele requires 2 copies to be expressed, otherwise, it remains hidden. Zygosity and chromosome sets. Homozygous, having 2 identical alleles for a specific gene. Heterozygous, having 2 different alleles for a specific gene. Diploid, cells that contain 2 sets of chromosomes, one from each parent. Haploid, cells that contain only one single set of chromosomes, such as sperm and egg cells. Inheritance patterns. Dominant versus recessive. An allele is dominant if it masks the effect of a recessive allele, e.g., brown eyes are dominant over blue. A recessive allele is only expressed when an individual inherits 2 copies of it, one from each parent. Homozygous versus heterozygous. An individual is homozygous if they have 2 identical alleles for a gene, and heterozygous if they have 2 different alleles. Mendelian inheritance. Based on Gregor Mendel's laws, this describes traits that are controlled by a single gene and follow predictable, straightforward patterns of dominant and recessive expression. The flow of genetic information. DNA replication, the process by which DNA copies itself exactly before a cell divides, ensuring each new cell has the same genetic instructions. Transcription, the process in the cell's nucleus where a specific segment of DNA is copied into messenger RNA, mRNA. Translation, the process in the cell's cytoplasm where mRNA is read by ribosomes to build a string of amino acids, which ultimately fold into functional proteins. Cell division. Mitosis, cell division that results in 2 identical daughter cells, responsible for growth and tissue repair. Meiosis, specialized cell division that produces reproductive cells, sperm and egg, cutting the chromosome number in half to ensure offspring have the correct number of chromosomes. Since the dawn of civilization, humankind has recognized the influence of heredity and applied its principles to the improvement of cultivated crops and domestic animals. A Babylonian tablet more than 6,000 years old, for example, shows pedigrees of horses and indicates possible inherited characteristics. Other old carvings show cross-pollination of date palm trees. Most of the mechanisms of heredity, however, remained a mystery until the 19th century, when genetics, as a systematic science, began. Genetics arose out of the identification of genes, the fundamental units responsible for heredity. Genetics may be defined as the study of genes at all levels, including the ways in which they act in the cell and the ways in which they are transmitted from parents to offspring. Modern genetics focuses on the chemical substance that genes are made of, called deoxyribonucleic acid, or DNA, and the ways in which it affects the chemical reactions that constitute the living processes within the cell. Gene action depends on interaction with the environment. Green plants, for example, have genes containing the information necessary to synthesize the photosynthetic pigment chlorophyll that gives them their green color. Chlorophyll is synthesized in an environment containing light because the gene for chlorophyll is expressed only when it interacts with light. If a plant is placed in a dark environment, chlorophyll synthesis stops because the gene is no longer expressed. Genetics as a scientific discipline stemmed from the work of Gregor Mendel in the middle of the 19th century. Mendel suspected that traits were inherited as discrete units, and although he knew nothing of the physical or chemical nature of genes at the time, his units became the basis for the development of the present understanding of heredity. All present research in genetics can be traced back to Mendel's discovery of the laws governing the inheritance of traits. The word genetics was introduced in 1905 by English biologist William Bateson, who was one of the discoverers of Mendel's work and who became a champion of Mendel's principles of inheritance. Although scientific evidence for patterns of genetic inheritance did not appear until Mendel's work, history shows that humankind must have been interested in heredity long before the dawn of civilization. Curiosity must first have been based on human family resemblances, such as similarity in body structure, voice, gait, and gestures. Such notions were instrumental in the establishment of family and royal dynasties. Early nomadic tribes were interested in the qualities of the animals that they herded and domesticated, and undoubtedly, bred selectively. The first human settlements that practiced farming appear to have selected crop plants with favorable qualities. Ancient tomb paintings show racehorse breeding pedigrees containing clear depictions of the inheritance of several distinct physical traits in the horses. Despite this interest, the first recorded speculations on heredity did not exist until the time of the ancient Greeks. Some aspects of their ideas are still considered relevant today. Hippocrates, c. 460 c. 375 BCE, known as the father of medicine, believed in the inheritance of acquired characteristics. And to account for this, he devised the hypothesis known as pangenesis. He postulated that all organs of the body of a parent gave off invisible seeds, which were like miniaturized building components, and were transmitted during sexual intercourse, reassembling themselves in the mother's womb to form a baby. Aristotle, 384 to 322 BCE, emphasized the importance of blood in heredity. He thought that the blood supplied generative material for building all parts of the adult body, and he reasoned that blood was the basis for passing on this generative power to the next generation. In fact, he believed that the male's semen was purified blood and that a woman's menstrual blood was her equivalent of semen. These male and female contributions united in the womb to produce a baby. The blood contained some type of hereditary essences, but he believed that the baby would develop under the influence of these essences, rather than being built from the essences themselves. Aristotle's ideas about the role of blood in procreation were probably the origin of the still prevalent notion that somehow the blood is involved in heredity. Today people still speak of certain traits as being in the blood, and of bloodlines and blood ties. The Greek model of inheritance, in which a teeming multitude of substances was invoked, differed from that of the Mendelian model. Mendel's idea was that distinct differences between individuals are determined by differences in single yet powerful hereditary factors. These single hereditary factors were identified as genes. Copies of genes are transmitted through sperm and egg and guide the development of the offspring. Genes are also responsible for reproducing the distinct features of both parents that are visible in their children. In the 2 millennia between the lives of Aristotle and Mendel, few new ideas were recorded on the nature of heredity. In the 17th and 18th centuries, the idea of preformation was introduced. Scientists using the newly developed microscopes imagined that they could see miniature replicas of human beings inside sperm heads. French biologist Jean-Baptiste Lamarck invoked the idea of the inheritance of acquired characters, not as an explanation for heredity, but as a model for evolution. He lived at a time when the fixity of species was taken for granted, yet he maintained that this fixity was only found in a constant environment. He enunciated the law of use and disuse, which states that when certain organs become specially developed as a result of some environmental need, then that state of development is hereditary and can be passed on to progeny. He believed that in this way, over many generations, giraffes could arise from deer-like animals that had to keep stretching their necks to reach high leaves on trees. Gregor Mendel, 1822 to 1884, an Austrian monk often called the father of genetics, discovered the fundamental laws of heredity. By crossbreeding thousands of garden pea plants in the 1850s and 1860s, he tracked specific traits like height or seed color across generations, proving that traits are passed down as distinct, discrete units, now known as genes. Mendel chose pea plants because they grow quickly, produce many offspring, and allow him to easily control pollination, the transfer of pollen from one flower to another. Before Gregor Mendel, theories for a hereditary mechanism were based largely on logic and speculation, not on experimentation. In his monastery garden, Mendel carried out a large number of cross-pollination experiments between variants of the garden pea, which he obtained as pure breeding lines. He crossed peas with yellow seeds to those with green seeds and observed that the progeny seeds, the first generation, F1, were all yellow. When the F1 individuals were self-pollinated or crossed among themselves, their progeny, F2, showed a ratio of 3 to 1, 3/4 yellow, and 1/4 green. He deduced that, since the F2 generation contained some green individuals, the determinants of greenness must have been present in the F1 generation, although they were not expressed because yellow is dominant over green. From the precise mathematical 3 to 1 ratio, of which he found several other examples, he deduced not only the existence of discrete hereditary units, genes, but also that the units were present in pairs in the pea plant and that the pairs separated during gamete formation. Hence, the 2 original lines of pea plants were proposed to be YY, yellow, and YY, green. The gametes from these were Y and Y, thereby producing an F1 generation of YY that were yellow in color because of the dominance of Y. In the F1 generation, half the gametes were Y and the other half were Y, making the F2 generation produced from random mating 1/4 YY, 1/2 YY, and 1/4 YY, thus explaining the 3 to 1 ratio. The forms of the pea color genes, Y and Y, are called alleles. Mendel also analyzed pure lines that differed in pairs of characters, such as seed color, yellow versus green, and seed shape, round versus wrinkled. The cross of yellow round seeds with green wrinkled seeds resulted in an F1 generation that were all yellow and round, revealing the dominance of the yellow and round traits. However, the F2 generation produced by self-pollination of F1 plants showed a ratio of 9 to 3 to 3 to 1, 9/16 yellow round, 3/16 yellow wrinkled, 3/16 green round, and 1/16 green wrinkled. Note that a 9 to 3 to 3 to 1 ratio is simply 2 3 to 1 ratios combined. From this result and others like it, he deduced the independent assortment of separate gene pairs at gamete formation. Mendel's success can be attributed in part to his classic experimental approach. He chose his experimental organism well and performed many controlled experiments to collect data. From his results, he developed brilliant explanatory hypotheses, and went on to test these hypotheses experimentally. Mendel's methodology established a prototype for genetics that is still used today for gene discovery and understanding the genetic properties of inheritance. Mendel's genes were only hypothetical entities, factors that could be inferred to exist in order to explain his results. The 20th century saw tremendous strides in the development of the understanding of the nature of genes and how they function. Mendel's publications lay unmentioned in the research literature until 1900, when the same conclusions were reached by several other investigators. Then there followed hundreds of papers showing Mendelian inheritance in a wide array of plants and animals, including humans. It seemed that Mendel's ideas were of general validity. Many biologists noted that the inheritance of genes closely paralleled the inheritance of chromosomes during nuclear divisions, called meiosis, that occur in the cell divisions just prior to gamete formation. It seemed that genes were parts of chromosomes. In 1910, this idea was strengthened through the demonstration of parallel inheritance of certain Drosophila, a type of fruit fly, genes on sex-determining chromosomes, by American zoologist and geneticist Thomas Hunt Morgan. Morgan and one of his students, Alfred Henry Sturtevant, showed not only that certain genes seemed to be linked on the same chromosome, but that the distance between genes on the same chromosome could be calculated by measuring the frequency at which new chromosomal combinations arose. These were proposed to be caused by chromosomal breakage and reunion, also known as crossing over. In 1916, another student of Morgan's, Calvin Bridges, used fruit flies with an extra chromosome to prove beyond reasonable doubt that the only way to explain the abnormal inheritance of certain genes was if they were part of the extra chromosome. American geneticist Hermann Joseph Muller showed that new alleles, called mutations, could be produced at high frequencies by treating cells with X-rays, the first demonstration of an environmental mutagenic agent. Mutations can also arise spontaneously. In 1931, American botanist Harriet Creighton and American scientist Barbara McClintock demonstrated that new allelic combinations of linked genes were correlated with physically exchanged chromosome parts. A major landmark was attained in 1953 when American geneticist and biophysicist James D. Watson and British biophysicists Francis Crick and Maurice Wilkins devised a double helix model for DNA structure. Their breakthrough was made possible by the work of British scientist Rosalind Franklin, whose X-ray diffraction studies of the DNA molecule shed light on its helical structure. The double helix model showed that DNA was capable of self-replication by separating its complementary strands and using them as templates for the synthesis of new DNA molecules. Each of the intertwined strands of DNA was proposed to be a chain of chemical groups called nucleotides, of which there were known to be 4 types. Because proteins are strings of amino acids, it was proposed that a specific nucleotide sequence of DNA could contain a code for an amino acid sequence, and hence protein structure. In 1955, American molecular biologist Seymour Benzer, extending earlier studies in Drosophila, showed that the mutant sites within a gene could be mapped in relation to each other. His linear map indicated that the gene itself is a linear structure. In 1958, the strand separation method for DNA replication, called the semiconservative method, was demonstrated experimentally for the first time by American molecular biologist Matthew Meselson and American geneticist Franklin W. Stahl. In 1961, Crick and South African biologist Sidney Brenner showed that the genetic code must be read in triplets of nucleotides, called codons. American geneticist Charles Yanofsky showed that the positions of mutant sites within a gene matched perfectly the positions of altered amino acids in the amino acid sequence of the corresponding protein. In 1966, the complete genetic code of all 64 possible triplet coding units for codons, and the specific amino acids they code for, was deduced by American biochemists Marshall Nirenberg and Hargobind Khorana. Subsequent studies in many organisms showed that the double helical structure of DNA, the mode of its replication, and the genetic code are the same in virtually all organisms, including plants, animals, fungi, bacteria, and viruses. In 1961, French biologist François Jacob and French biochemist Jacques Monod established the prototypical model for gene regulation by showing that bacterial genes can be turned on, initiating transcription into RNA and protein synthesis, and off through the binding action of regulatory proteins to a region just upstream of the coding region of the gene. Here is the timeline of important milestones in the history of genetics. In 1866, Austrian botanist Gregor Mendel published the results of his experiments with pea plants. His work later provided the mathematical foundation of the science of genetics. In 1869, Swiss biochemist Johann Friedrich Miescher became the first to isolate nuclein, now known as DNA. Although he developed hypotheses explaining the role of nuclein in heredity, he ultimately concluded that one molecule alone could not provide the level of variation observed in nature within and between species. In 1900, Mendel's experiments were rediscovered independently by Dutch botanist and geneticist Hugo de Vries, German botanist and geneticist Carl Erich Correns, and Austrian botanist Erich Czermak von Ciesneg, giving rise to the modern science of genetics. In 1928, English bacteriologist Frederick Griffith conducted experiments suggesting that bacteria are capable of transferring genetic information, and that such transformation is heritable. In 1931, American scientists Harriet B. Creighton and Barbara McClintock published a paper demonstrating that new allelic combinations of linked genes are correlated with physically exchanged chromosome parts. Their findings suggested that chromosomes form the basis of genetics. In 1944, Canadian-born American bacteriologist Oswald Avery and American biologists Macklin McCarty and Colin MacLeod reported that the transforming substance, the genetic material of the cell, was DNA. In 1950, Austrian-born American biochemist Erwin Chargaff discovered that the components of DNA are paired in a 1-to-1 ratio. Thus, the amount of adenine, A, is always equal to the amount of thymine, T, and the amount of guanine, G, is always equal to the amount of cytosine, C. In 1951, British scientists Rosalind Franklin, Maurice Wilkins, and Raymond Gosling conducted X-ray diffraction studies that provided images of the helical structure of DNA fibers. In 1953, using Chargaff's data and the X-ray images recorded by Franklin, Wilkins, and Gosling, British biophysicists James Watson and Francis Crick determined the molecular structure of DNA. Watson, Crick, and Wilkins shared the 1962 Nobel Prize for Physiology or Medicine for their discovery. In 1960s, Swiss microbiologist Werner Arber and American microbiologists Hamilton Othniel Smith and Daniel Nathans discovered restriction enzymes, which cleave DNA into fragments. The discovery, for which the three men shared the 1978 Nobel Prize for Physiology or Medicine, enabled scientists to manipulate genes by removing and inserting DNA sequences. In 1970s, American molecular biologists Alan M. Maxim and Walter Gilbert and English biochemist Frederick Sanger developed some of the first techniques for DNA sequencing. Gilbert and Sanger shared the 1980 Nobel Prize for Chemistry for their work. American biochemist Carey B. Mullis invented the polymerase chain reaction, PCR, a simple technique that allows a specific stretch of DNA to be copied billions of times in a few hours. Mullis received the 1993 Nobel Prize for Chemistry for his invention. The Human Genome Project, HGP, began. By the time of its completion in 2003, HGP researchers had successfully determined, stored, and rendered publicly available the sequences of almost all the genetic content of the human genome. The International HapMap project, which was designed to identify genetic variations contributing to human disease through the development of a haplotype, haploid genotype map of the human genome, began. By completion of Phase 2 of the project in 2007, scientists had data on some 3.1 million variations in the human genome. The 1000 Genomes Project, an international collaboration in which researchers aim to sequence the genomes of a large number of people from different ethnic groups worldwide with the intent of creating a catalogue of genetic variations, began. The project was completed in 2015. Genetics is the study of heredity and the variation of inherited traits. It rests on a few foundational principles that explain how biological information is stored, passed down, and expressed across all living organisms. Let's explore the following core concepts of genetics for a clear understanding of the field. DNA, genes, and chromosomes are the fundamental components that make up the human instruction manual. They can be visualized as an organizational hierarchy. DNA makes up genes, and genes are tightly packaged into chromosomes. Deoxyribonucleic acid, DNA, is the master molecule of life, carrying the hereditary blueprint and biological instructions required for the development, survival, and reproduction of nearly all living organisms. It acts like an ultimate instruction manual or cellular recipe book, telling your cells exactly how, what, and when to build crucial molecules like proteins. In humans, DNA is distributed among 46 chromosomes nestled inside the protective walls of the cell nucleus, with tiny fractions also living in your energy-producing mitochondria. DNA inside a human cell acts as a master instruction manual. Most is tightly packaged inside the nucleus as 23 pairs of chromosomes, while a small amount sits in the mitochondria. If completely unraveled, the DNA from a single cell would stretch about 2 meters long. Now let's understand the molecular structure of DNA. DNA is a long polymer made up of repeating subunits called nucleotides. To understand what DNA looks like, imagine a long, winding ladder twisted into a spiral shape known as a double helix, formed by two complementary strands of nucleotides that run antiparallel to each other and are held together by hydrogen bonds. Every single strand of DNA is a polymer made up of repeating monomer units called nucleotides. Each nucleotide consists of 3 distinct chemical parts. Deoxyribose sugar, a 5-carbon pentose sugar molecule. Phosphate group, a chemical group attached to the 5' carbon of the sugar. Nitrogenous base, a nitrogen-containing molecule attached to the 1' carbon of the sugar. The 4 nitrogenous bases found in DNA are adenine, A, thymine, T, cytosine, C, and guanine, G. In RNA, the base uracil, U, replaces thymine. These nitrogen-containing organic molecules serve as the fundamental alphabet of the genetic code. Nitrogenous bases are divided into 2 distinct chemical groups based on their ring structures. Purines, double ring structure, consist of a 6-membered ring fused to a 5-membered ring. Adenine, A, found in both DNA and RNA. Guanine, G, found in both DNA and RNA. Pyrimidines, single ring structure, consist of a single 6-membered ring. Cytosine, C, found in both DNA and RNA. Thymine, T, found only in DNA. Uracil, U, found only in RNA. In the structure of nucleic acids, bases pair up specifically via hydrogen bonds to stabilize the strands. In DNA, adenine pairs with thymine, A equals T, via 2 hydrogen bonds, while cytosine pairs with guanine, Cg, via 3 hydrogen bonds. Due to their unique molecular structures, bases always partner with specific counterparts, like a lock and key, held together by hydrogen bonds. DNA is a polymer of the 4 nucleotides A, C, G, and T, which are joined through a backbone of alternating phosphate and deoxyribose sugar residues. These nitrogen-containing bases occur in complementary pairs, as determined by their ability to form hydrogen bonds between them. A always pairs with T through 2 hydrogen bonds, and G always pairs with C through 3 hydrogen bonds. The spans of A, T and GC hydrogen bond pairs are nearly identical, allowing them to bridge the sugar phosphate chains uniformly. This structure, along with the molecule's chemical stability, makes DNA the ideal genetic material. The bonding between complementary bases also provides a mechanism for the replication of DNA and the transmission of genetic information. In 1953, James D. Watson and Francis H.C. Crick proposed a three-dimensional structure for DNA based on low-resolution X-ray crystallographic data collected by biophysicists Rosalind Franklin and Maurice Wilkins, and on Erwin Chargaff's observation that, in naturally occurring DNA, the amount of T equals the amount of A and the amount of G equals the amount of C. Watson and Crick, who shared a Nobel Prize in 1962 for their efforts, postulated that two strands of polynucleotides coil around each other, forming a double helix. The two strands, though identical, run in opposite directions as determined by the orientation of the 5'3" phosphodiester bond. The sugar phosphate chains run along the outside of the helix, and the bases lie on the inside, where they are linked to complementary bases on the other strand through hydrogen bonds. The double helical structure of normal DNA takes a right-hand form called the B helix. The helix makes one complete turn approximately every 10 base pairs. bDNA has two principal grooves, a wide major groove and a narrow minor groove. Many proteins interact in the space of the major groove, where they make sequence-specific contacts with the bases. In addition, a few proteins are known to make contacts via the minor groove. Several structural variants of DNA are known. In aDNA, which forms under conditions of high salt concentration and minimal water, the base pairs are tilted and displaced toward the minor groove. Left-handed zDNA forms most readily in strands that contain sequences with alternating purines and pyrimidines. DNA can form triple helices when two strands containing runs of pyrimidines interact with a third strand containing a run of purines. bDNA is generally depicted as a smooth helix, however, specific sequences of bases can distort the otherwise regular structure. For example, short tracks of a residues interspersed with short sections of general sequence result in a bent DNA molecule. Inverted base sequences, on the other hand, produce cruciform structures with four-way junctions that are similar to recombination intermediates. Most of these alternative DNA structures have only been characterized in the laboratory, and their cellular significance is unknown. DNA does not simply sit idly in the cell, it drives the daily mechanics of life through three critical roles. The unique, sequential order of A, T, C, and G serves as an alphabet. Specific stretches of this code form genes, which are recipes for making proteins. Strikingly, genes only make up about 1% of human DNA, the rest regulates how and when those genes are used. The unique, sequential order of A, T, C, and G serves as an alphabet. Specific stretches of this code form genes, which are recipes for making proteins. Strikingly, genes only make up about 1% of human DNA, the rest regulates how and when those genes are used. DNA dictates physical traits by sending messages. It undergoes transcription, where a gene sequence is copied into a temporary messenger molecule called RNA. This RNA is then read by the cell to assemble amino acids into proteins. The fundamental physical and functional unit of heredity. Genes are specific segments of DNA that contain the instructions needed to build proteins, which perform most cellular functions. A gene is the basic physical and functional unit of heredity, made up of sequences of DNA. Acting as a precise set of instructions, genes tell cells how to make specific molecules, such as proteins, that dictate everything from how the body functions to physical traits like eye color. Arranged sequentially on structures called chromosomes, which are housed inside the cell nucleus. Small numbers of genes also exist in the mitochondria. The human body contains an estimated 19,900 to 20,000 protein-coding genes. These instructions account for less than 2% of the entire human genome. Humans inherit 2 copies of each gene, one from each biological parent. Variants of the same gene are known as alleles, which account for different physical traits like eye color or blood type. Genes achieve their effects through a process called gene expression. During expression, the DNA sequence is first transcribed into ribonucleic acid, RNA. For protein-coding genes, This RNA serves as a blueprint to assemble amino acids into proteins that drive biological functions, structure tissues, and dictate physical traits. Non-coding genes produce functional RNA molecules that directly help regulate and control other genes. The process of transcription and translation will be discussed in detailed videos in the future. Long, thread-like structures composed of DNA and proteins. Humans typically have 46 chromosomes, 23 pairs, in almost every cell, inheriting one set of 23 from each parent. A chromosome is a tightly packaged, thread-like structure of DNA and proteins found inside the nucleus of most living cells. It carries the primary genomic information necessary for organism development, survival, and reproduction. Chromosomes possess a distinct structural arrangement that protects and organizes genetic material. DNA double helix, the fundamental genetic material carrying hereditary instructions. Histons, specialized packaging proteins around which DNA tightly coils. Chromatin, the combined DNA-protein complex that condenses to form a chromosome. Centromere, the constricted central point dividing the chromosome into two arms. P-arm, the short upper arm of the chromosome structure. Q-arm, the long lower arm of the chromosome structure. Telomeres, repeating DNA caps at the ends protecting the structure from damage. Chromosomes are classified into four primary types, based on the position of the centromere, the constricted region that divides the chromosome into two arms. Metacentric, the centromere is located right in the center, creating two arms of roughly equal length. This gives the chromosome a distinct V-shape. Submetacentric, the centromere is slightly offset from the center, resulting in one shorter arm, P-arm, and one longer arm, Q-arm. This gives the chromosome an L shape or J shape. Acrocentric, the centromere is located very close to one end, creating one extremely long arm and one very short, knob-like arm. Telecentric, the centromere is situated at the very tip or terminal end of the chromosome. These chromosomes have only one arm and are rod-shaped. They do not occur in humans, but are found in other species like mice.